In the second part of this week's lecture, we will now start to look in more detail at DNA methylation and post-translational modifications of histones. Firstly, DNA methylation. What we see in this diagram is a picture of a promoter and an enhancer. Shown bound to the promoter and the enhancer are numerous transcription factors. We also see there are sites of potential DNA methylation shown here as these CpG dinucleotides going from the 5' to the 3' direction. This is the normal symbol for indicating methylated Cs at CpG dinucleotides. You can see that when the promoter is unmethylated, we have a less condensed form of chromatin and genes can be transcribed. However, if DNA is methylated, shown here with this closed-in lollipop symbol, it can inhibit the binding of transcription factors and of RNA polymerase II, shutting down gene transcription. In this case, the chromatin takes on a much more condensed form, as we see here. Histones. Histones are made up of 8 subunits. We can see that the core histone is an octamer of 4 subunits, 2 times H2A, 2 times H2B, 2 times H3, and 2 times H4. The DNA itself is wrapped around this histone octamer that we see here. Protein tails of the histone subunits can protrude out, and the numbers, such as this, indicate amino acid residues which can be modified. For example, those numbers in red can be acetylated and they are lysines, whereas in black we have arginines that can be methylated, shown here. And you can see there is also methylated lysine, shown in blue, as well as ubiquitinylation and phosphoserine. So histone subunits can be highly modified by methylation, acetylation, and phosphorylation, as well as ubiquitinylation, all of which are important for understanding epigenetics. There are 2 types of enzymes which are important in this modification of histones. There is a histone acetyltransferase, or HAT, which transfers an acetyl group onto histone, helping the transition from closed to more open chromatin. The reverse enzyme is called a histone deacetylase, or HDAC. Different marks of histones are associated with differences in expression. Histone acetylation, for example, is associated with very high levels of gene expression. The acetylation of the histones changes the charge of the histone and loosens the interaction between the DNA and the histone. What we see in this figure here is information on the methylation of histones. And in this case, we're looking at, for example, lysine residue 4 on histone 3. These plots are what we call microarrays. They are arranged in a series of columns and rows. The color in these columns and rows, for example here, if it's dark blue, it indicates high expression of a particular gene. Regions that are light blue indicate very low expression. This is also known as a heat map. Hot colors, high expression; cooler colors, low expression. What we can see is that expressed genes have a mark called histone lysine 4 methyl 3. This is a trimethyl group. And you can see a very large number of these highly expressed genes in dark blue. When genes are turned off, shown here on the right-hand panel, you can see that these many genes are either repressed or turned off completely. There is also a signal for turning off genes, and that's shown here. So for example, lysine 27 trimethylation is absent from genes that are turned on, whereas when genes are turned off, we see an increase in the amount of lysine 27 trimethylation on those same genes. So in other words, what these two figures have shown is that changes in the amount of methylation at specific residues of, in this case, histone 3, are correlated with genes being turned on, lysine 4 trimethylation, or genes being turned off, lysine 27 trimethylation. So specific histone methyltransferases and demethylases, these are enzymes that either add or take away methyl groups, catalyze the complex methylation pattern of histones. And this is critical for regulating gene transcription. But what about the intersection between epigenetics and genetics? Rare mutations and polymorphisms are found in both promoters and enhancers and can exert an effect on gene expression. This change in gene expression reflects changes in the epigenetic modification of DNA and histones. So how does that work? What we have here, again, is our enhancer and our promoter. Within the promoter is a sequence, shown here, and you can see that in that sequence, there are two CG dinucleotides moving from the 5' end. This methylation of the cytosine residue creates a binding site for a methyl binding protein. This methyl binding protein can in turn recruit other modifying enzymes, such as a histone deacetylase, removing the acetyl groups from histone, and a histone methyltransferase, acetylating, for example, histone 3 lysine 27. So DNA methylation provides a chemical signal allowing proteins to bind, which recruit further proteins, changing the modification of histones to favor gene repression and chromatin condensation. And that's shown. But what would happen if we had a mutation? That's shown in this diagram here. Now, for example, if this C residue was mutated, this would decrease the methylation of the promoter. This would have an effect on the methyl binding protein, which could then have an effect on the recruitment of histone modifying enzymes, thereby changing patterns of gene expression. Alternatively, a mutation may occur in the methyl binding protein itself, meaning that it was either unable to bind to DNA, or if it did bind to DNA, it exerted no repressive effect. So in other words, DNA methylation is a repressive mark in the context of gene expression. It represses gene expression through the recruitment of methyl binding proteins, which in turn recruit histone deacetylases, as well as specific histone methyltransferases. So let's move on to an example. Rett syndrome is an X-linked neurodevelopmental disorder. This means it is due to a mutation in a gene carried on the female X chromosome. It is lethal in males. Males have only one X chromosome, and if this gene is mutated, then that is lethal. The children, or the girls, start out normal, and then between 6 and 18 months of age, develop severe neurological abnormalities. Despite this, the affected girls can survive into middle age, and it affects between 1 in 10 and 20,000 girls worldwide. The gene which carries a mutation in Rett syndrome is called MECP2, and it encodes a methyl binding protein. So MECP2 silences gene expression by binding to methylated DNA, recruiting a histone deacetylase, and resulting in chromatin remodeling, such as we see here. In the presence of neuronal activity, however, the gene is turned on and methyl CP2 is lost from the chromatin. allowing transcription of a target gene. So in effect, what we see is that MECP2 is critical for regulating gene expression in response to neuronal activity. You could imagine that the loss of MECP2 increases the number of genes that are turned on in the absence of neuronal activity. So it dysregulates critical neuronal pathways early in development. What this table here shows is the genetic heterogeneity of the MECP2 gene. We've already seen this before in cystic fibrosis. Here are all of the mutations that have been characterized in European populations. You can see that in this case, they are all single nucleotide changes, for example, 27G to a T. And in this case, it changes an arginine to a serine. What you will also see is that there are a number of mutations here and here and here, which are nonsense mutations. In other words, the mutation has led to the introduction of a stop codon shown by an asterisk. So MECP2 is a methyl binding protein that turns genes off. Loss of this gene changes patterns of gene expression during a critical period of brain development and activity. Fragile X is another example of a disease caused by dysregulation of epigenetic programming. This particular disease is related to Huntington's disease in that it is an expansion of triplet repeats. However, unlike Huntington's disease, it is an expansion of a triplet repeat in the 5' untranslated region. In other words, it's happening up near the promoter. And it is an expansion of a repeat CGG. And you can immediately see that there is a 5' C which could become methylated. In the population, it is highly polymorphic. Anywhere between 6 and 54 alleles is normal. However, the unstable repeat number can be anywhere between 200 and 1,000 repeats. So it's a huge expansion. The expansion of this triplet repeat causes the structure of DNA to change such that it can be easily identified under a light microscope that we see here. So, very, very large number of repeats. Compare and contrast this to Huntington's disease, where we saw a much lower number of repeats associated with the disease state. Fragile X is due to the expansion of this CGG repeat in a gene called FMRP. You can see the methylation is taking place near the promoter of this gene in what's called a CPG island, a highly CPG-rich area of the gene. It is normally either not methylated or methylated with very low frequency, and an increasing number of these repeat sequences increases methylation, shutting down gene expression. So what we can see is that in the normal situation, the gene is turned on. We see acetylation of the histone subunit, shown here, for example, whereas expansion of this triplet repeat, shown here, leads to loss of histone acetylation, the recruitment of MECP2, the protein we've already talked about in the context of Rett's disease, the contraction of chromatin, and the shutting down of gene expression. So in summary, Fragile X is similar to Huntington's disease in that it is due to the expansion of a trinucleotide repeat. While Huntington's disease is caused by the expansion of a CAG repeat that increases the number of glutamine residues in a protein, Fragile X increases a CGG repeat, which increases the DNA methylation of the FMR1 gene. Repression of the FMR1 gene by this increased DNA methylation results in the loss of a protein that is otherwise essential for normal cognitive development.